for this nice introduction. Indeed, I will tell you something more about the detection of microbial contamination in food production and targeted cleaning and disinfection. I'm looking, yeah, okay, next slide. So if we look to the top 10 of the causes of foodborne outbreaks in the United States, well, then we see that on the third place, so the third most important cause of a foodborne outbreak is in fact the insufficient cleaning of apparatus used to produce those food products. Besides also, if we look to literature, and not only literature, also experience at ILVO, we see that a lot of food companies have problems with persistent contaminations with foodborne pathogens, like, for example, Listeria monocytogenes. So, and that contamination with Listeria monocytogenes can take up till more than 10 years. So it's clear that cleaning and disinfection plays an important role in food production. And here you see the roadmap for cleaning and disinfection, and you see that in fact in that roadmap you have at least 12 steps. And beside those 12 steps, well, in between, you have to check if the cleaning and disinfection is well performed. And there are also several stages in which you can check that. One of the important stages is, of course, after cleaning. After cleaning with the product followed by rinsing, well, it's needed to do visual control. We have some problems with the slides. Yeah, okay, a visual control. So, and beside that visual, to look if all the dirtiness is away, and beside that visual control, I always advise the companies to also do at least some biochemical tests, like, for example, ATP measurements, because by doing ATP measurements, you can look if still organic matter is present. And a second check is, in fact, after your disinfection. There you need to do microbial analysis to check if the disinfection is well done. Now, concerning those microbiological tests, you have in fact two types of tests you can perform, or you can use contact methods, or you can use swab methods. Now, if you use contact methods, then in fact you are more focused on the enumeration of hygiene indicators. Hygiene indicators like total aerobic flora, Enterobacteriaceae, yeast and molds. And there are different contact methods you can use, like you see also in the pictures, or you can use an agar plate, or you can use agar slides in the middle, or you can use a Petri film. And beside contact methods, also swab methods can be used, but those methods are of course more focused on the pathogen detection, also looking for pathogen absence after cleaning and disinfection. Now, if you use swabs, of course, you need to use the correct swab at the correct moment. So you have a lot of types of swabs which can be used, but if you have to deal with hard to reach points, which also have a small area, so less than 25 square centimeters, then you need to use the environmental swabs on the left-hand side of the slide. If the area is bigger, well, then you need to use the sticks. And if you can sample more than 1,000 square centimeters, then sponges or gauze swabs need to be used. Then, of course, the next thing is which kind of points do I need to sample? How many points do I need to sample? At which frequency? And which type of microorganisms do I need to analyze? Well, concerning the sampling points, well, for hygiene control, well, those sampling points need to be risk-based. And beside the fact that they need to be risk-based, of course, you also have to give attention to wet points, soiled points, and certainly also hard to reach and poorly cleanable points. And the area, well, I always say, if you are looking for pathogen, please swap areas as big as possible. So if you can, well, take 3,000 square centimeters. We at ILVO even try to sample half a square meter if possible. Of course, for your hygiene indicators, the surface is smaller because you need to use there your contact methods and use your contact devices, and they fix you the area which is available for that kind of method. Then, how many samples do I need to take? At which frequency and which type of microorganisms? Well, this is depending on the zone you are sampling. So if you are sampling zone 1 points, so food contact surfaces, of course, then you need to sample most places. Also at the highest frequency, so in my opinion, daily. And concerning the type of microorganisms, well, beside pathogens, it's also useful there and advisable to sample for some hygiene indicators. While, for example, if you are looking for zone 3 points, so non-food contact surfaces, which are not so close to zone 1, so like a wall, a floor, or a drain, well, then, for example, sampling for pathogen detection can be enough. And for example, on a weekly basis can also, in a lot of cases, be enough. Then please give absolute attention to difficult to reach and difficult to clean and disinfect food contact surfaces and places. A slicing machine is really a source of pathogen and hygiene contamination. So if you have slicing machines, please take the time to disassemble those slicing machines and clean and disinfect them very profoundly. For example, also an infusion room of UHT, like you can see here, even after sip, it's needed to do a disassembling because you see that even after sipping, so still dirtiness is there. And so only with disassembling the infusion room, you can clean and disinfect the infusion room in a proper way. And then, of course, also your cleaning tools. Your cleaning tools you use, even after cleaning and disinfection of your cleaning tools, it can be a source of contamination. In some studies, and also at ILVO we see that, in some studies we see that although your cleaning tools are cleaned and disinfected and put away for the next day, that up till 70% of those cleaning tools can still contain Listeria monocytogenes. At ILVO, we also guide and help companies to get rid of their contaminations. But the slides give some delay. Okay, no. First, a sampling moment. On which moments do you now have to sample? Well, that's depending on what you want to check. If you want to check your cleaning and disinfection, of course, you need to sample after the cleaning and disinfection. But we always advise to not sample immediately after cleaning and disinfection, but just before start up of your production. Because at the moment of start of your production, there are some hours between the cleaning and disinfection and the start of up of your production. And during that period, there can be some, recontamination. So it's advisable to sample before start of production to check your hygiene before you start your production. If you want to know, for example, the problems with Listeria during processing, well, then you can do that also, and you can sample your environment and your production line also during processing for pathogen detection, but then at least sample only after at least 2 hours of production. Like I told you before, so we also guide a lot of food companies in getting rid of their problems with, for example, Listeria monocytogenes contamination. And here you have an example where we work together with a meat processing plant, and it took up till more than 9 months to get rid of their problems with Listeria monocytogenes. You see, in fact, that at the start, even after cleaning and disinfection, still 50% of the points that we sampled contained Listeria monocytogenes, and after 9 months intensive work, corrective actions, and so on, we could end at the contamination of only 7% in the environment after cleaning and disinfection. And then the last aspect I want to show to you is that it's only also sometimes very interesting to type your Listeria monocytogenes strains. So to look if you have Listeria monocytogenes, if you have to deal with one persistent strain or a zoo of different strains. And so at ILVO, we do fingerprinting. So we make the fingerprint of all Listeria monocytogenes isolates. And you see in that company, we had 5 different types being present in that company. And during the first sampling, you see that, in fact, especially the orange strain was persistently present as well in the raw area as in the cooked area, 7 times in the cooked area, 3 times in the raw area, and even in the packaging area, which is even in high care area. So which is of course not done, having Listeria in your high care area. But you see that the dominating orange strain could enter also in the packaging area. But what you can also see is that the light blue strain was dominating during the second sampling and also entering the packaging area 2 times. And that strain was also already available there or present there as the second dominant strain 2 weeks before. So we can say that that light blue strain is really a persister. And so by typing your Listeria monocytogenes strains, you can also look for contamination, sources, routes, and persistence. So this is the end of my presentation. Of all those things I just mentioned to you, we can give you, of course, scientific support. And so do not hesitate to contact us, and I will be very happy to answer some questions.
So thank you very much, Koen Rue, for your very clear talk and your very interesting topic. I will have a look at Natalie and Kathleen. Are there any questions for Koen?
Yes, indeed, there are some questions in the chat. For example, we have a question. For Koen de Reu, what's your point of view on screening for Listeria species as an environmental tool? Do you have an idea of this?
Yeah, I think this is a very good idea because, okay, you have the Listeria species and global, and only Listeria monocytogenes is pathogenic. But it's indeed a very good idea, and a lot of companies also do that, to monitor for Listeria species, because if you see that Listeria species is already in your food production environment, then there is a big chance that one day or another day Listeria monocytogenes will appear in your food production. So it's indeed a very good idea to get rid from your Listeria species. Then you're sure that you don't have Listeria monocytogenes. On the other hand, in some food companies, that this can be too much asked. So if, for example, in meat processing plants, you have a huge number of Listeria species entering, so sometimes it's really not possible to get rid of the Listeria species, and they need to focus on monocytogenes. But if possible, please indeed try to get rid of your Listeria species already. And thanks for the good question.
Okay, we have some more questions. If you sample before, during and after production, how long do you need to wait until we get the result of the microbial loads? You can estimate this?
Well, this is depending on the lab, of course, who is doing the analysis. So as we do ISO methods, this takes a little bit longer. Mostly we help companies in a framework of a research project, and this takes then a little bit longer because we really use the reference method, but also shorter methods, and we also can do that, but then we don't have an accreditation therefore, but that's not always needed. But so normally you can already have your result within 48 hours. Even if it's absent, you can already have your result within 36 hours. There are also sometimes some really very short tests which you can perform by yourself, only based on colorations, and you can incubate them by yourself, but those tests are really not so trustful.
Okay, thanks, Koen. So you mentioned, Koen, that you need to swap 1,000 to 3,000 square centimeter of area, but this seems a bit large. Is, for example, 100 square centimeter also enough, or is the area, does it need to be big enough?
First of all, I'm very happy, of course, Simba, that you have a question for me. So yes, I know in the kind of production you work, Simba, but indeed, our advice is always to try to sample an area as big as possible. Indeed, in certain cases, it's not possible. And then you need to sample a smaller part, but even then try to sample as large as possible. What is also interesting, if you do a sampling of the environment, please, for each type of point, try to sample always an area which is as big as the days before, so that you can do a kind of, how do you call it, also trend analysis. So try to fix your areas for the types of sampling points so that you can have a history of standardized sampling. But of course, 100 square centimeters is enough in some cases, but to be honest, this is small. So then it must be really very small, how do you call it, sections of your production line. But if you can sample larger areas, please do that.
Okay, thank you, Koen. That was a very clear answer, and thank you all of you for asking your questions.